This conference will now be recorded.
Hello students, I'm Dr. Divya from TeenEinstein. Now we are in chapter 11, Biotechnology, Principles and Processes. So well, in the first class, that means part 1, we were discussing about what is biotechnology or how and for what and all we can use this or how we can combine biology and technology. And by combining this biology and new technologies, how to make a new or how to give a new approach like in vitro fertilization, test tube baby and all. For that new concepts, how we are giving a new form for that biology or biological approaches, how it is going to change. So all these things we were discussing. Then we discussed about principles of biotechnology, and we discussed the basic idea of genetic engineering and bioprocess engineering and all. Then we learned about tools of rDNA technology. rDNA technology means recombinant DNA technology. So how we are combining that recombination, different combinations of DNA, and what change that makes, or how that new technology is useful. So that and all we are learning in this chapter. So then we learned about the tools of rDNA technology, and which are all the key tools. Just generally we said about the tools: restriction enzymes, then polymerase enzyme, ligase, vectors, then host organism. Generally we said about all these things, which are all the key tools: restriction enzyme, polymerase enzyme, ligase, that is also another enzyme, then vectors, then host organism. All this we have discussed in last class. Then in today's class, we are going to discuss everything in detail. So we are moving to the deeper part of principles of biotechnology. This chapter is very, very important because this is new. All these concepts are new to you people. So it will be difficult to understand without knowing the basic concept. That's why I asked you, you should know about the tools of rDNA technology. Otherwise, you won't be able to understand the different processes which we are going to explain later. How to apply this. While applying all these technologies, you should be able to get that idea, how it is or why, for what purpose you are using that tool in that place. So you should have a clear idea, the role of each one. You should have a clear idea about the role. Okay. So first we are going to tell the 3 basic steps. For genetically modifying an organism, there are 3 basic steps. See, for organism, we need, why we are using this biotechnology? To change or to make modifications in the existing organism, right? If we need more production, we need to change something. We need to change the normal organism into a modified one. So we have to change the genome. We have to make the changes in the genome, or genetic material should be changed. Otherwise, definitely it will give the same thing only, right? A human being, if the genome structure is changing, that shows some kind of changes in next generation, right? Otherwise, the same thing will get repeated. For that, we will say cloning, the same thing. For repeating the same thing, for making more number of the same things, that is called as cloning. So we need some kind of modification. Genetically, we need modifications. The genetic material should be modified, or that should be changed. So for that, there are 3 steps. Basically, there are 3 steps So for genetical modification of an organism, first we need to identify the DNA with desirable gene. Okay. What do you mean by that? Desirable gene, whichever gene we want or whichever character should be improved, that gene first we need to find out the gene, that we need to locate that gene. We need to find out the position of that gene in the genome. So first thing or the first step is the identification of DNA with desirable gene. Which gene is the desirable one, or which one we want, which character we want to improve or which character we want to modify, that gene we need to find out. So identification of the DNA, whichever gene we need to identify that DNA with that desired gene. That desired gene on which DNA it is present, that we need to identify first. Clear? So first step is the identification of DNA with desirable gene. So whichever gene is desirable or which gene is desirable, which one you want, that DNA you need to find out. The position of that gene you should find out. So that is the first step. Then second step, first you are identifying or finding out the desirable gene. That is the first step. Then introduction of the desired or the identified DNA into host. After identification, what is the next step? Next step is the introduction of the identified DNA into host. So we need that in more number, isn't it? If you are identifying that gene, we need that in more number. We need to make modifications. So what we need to do for that? For multiplication, or if we need more number, we need to introduce. We need to introduce that identified DNA into a suitable host, right? We need to identify that into a suitable host. Okay. So the second step is introduction of the identified DNA into the host. First, for modification, first we need to find out the desirable gene, the DNA with desirable gene. Then that DNA with desirable gene should be introduced into a host. Okay. And the third step is maintenance of that DNA in that host. And while multiplication of that host, that DNA along with that gene, this desired gene also, that also will get multiplied, right? So it will get transferred into the progeny of that host. Got it? Don't get confused. First we need to identify the gene. That gene means not only that gene, we can identify the gene in DNA. That DNA with desirable gene. On that DNA, desirable gene is present or not, that we can confirm and we can identify that. Then we are introducing that particular part of DNA with that particular gene which we want, that we are introducing into a host. Okay, we are introducing that into a host. Then maintenance of introduced DNA. Then we need to maintain that introduced DNA, that DNA part or the DNA with desirable gene that is introduced into a host now. That should be maintained. So in that host, it is transferred into a host, and in that host DNA, along with the host DNA, that also will multiply. Okay, so after multiplication, what will happen? That will get transferred to the progeny. That DNA with desired gene, that will get transferred into the progeny of the host. So there are three steps: identification of the DNA with desirable gene, then introduction of the identified DNA into host, then third step, after introducing into host, maintaining that DNA along with the multiplication of host genome. This DNA also will get multiplied, then it will get transferred along with that host genome, that also will get transferred into the progeny. Got it? So these are the three main steps. Then we have told, or we have already discussed about this rDNA or recombinant DNA technology. We can perform only with the help of key tools. Key tools. Which are the key tools? What we said about the key tools: restriction enzymes, polymerase enzyme, ligases, vectors, Then host organisms. These are the important things. Restriction enzyme, polymerase enzyme, ligases, vectors, host organism. Okay. So all these things, all these are the key tools. Restriction enzyme, polymerase enzyme, ligase enzyme, vector and host organism. These are the key tools for our DNA technology. Then what is the role of each? We should know the role of which I said already. If you are not having any idea about the particular role of this restriction enzyme, polymerase enzyme, ligase, vector, host organism, we won't be able to learn anything in this chapter. So we have to get a clear idea about the role of each one. Okay, each tool. What is the role of each tool? So first we are going to learn about restriction enzyme. Just I will explain this and all already we have told. So restriction enzyme, what this restriction enzyme will do? This restriction enzyme will cut the double-stranded DNA. Okay, at the specific site, recognition site, this what this DNA restriction enzyme will do? This restriction enzyme that will identify particular cutting sites or that will recognize some sites, specific recognition sites will be there. On that point, this enzyme cuts the double-stranded DNA. Okay, that is the role of restriction enzyme. First, it will identify specific recognition site, and on that side, this enzyme cuts the double-stranded DNA. And this eliminates the infecting organism. Okay, actually, this enzyme is found in bacteria. Restriction enzyme that is found in bacteria, and that eliminates the infecting organism also. So what is the role of restriction enzyme? The role of this restriction enzyme that will identify the recognition site, specific recognition site that will identify, and on that site this restriction enzyme makes cuts and that cuts the double-stranded DNA in the specific site. Got it? Next, second one is polymerase enzyme. Okay, polymerase enzyme, DNA polymerase we'll say. So the polymerase enzyme. So this, what is the role of polymerase enzyme? Polymerase enzyme synthesize nucleotides. Okay, that is synthesizing nucleotides. And these nucleotides that will be complementary to the template strand. Okay. For the template strand, while this reaction, it should synthesize new strand. Right? While learning about PCR and all, we have learned. Okay. So synthesizing or DNA replication type and all, we know all these things. Polymerase enzyme, what is the role? It is polymerizing, that is helping for polymerization. Okay, so that nucleotide, synthesizing nucleotide which are complementary to the template strand. If there is a template strand, this polymerase enzyme that is helping to synthesize the complementary strand. Okay, if there is one strand, double-stranded DNA, it is separating. Then again, to synthesize the new strand, opposite strand, this polymerase enzyme is helping. How it is helping? It is synthesizing nucleotide which are complementary to the template strand. Template strand is the strand which is helping to, which is present there, and the opposite strand will be synthesized. Newly synthesized strand will be there. Okay. So in that strand, for synthesizing the complementary strand, DNA polymerase is helping. How it is helping? That is adding the nucleotides. Okay, to that free OH end, that is adding the nucleotide. And it is helping in the elongation of strand. Okay, for the elongated strands, it is helping. That means it is helping for continuous synthesis of that strand. Now we have learned about restriction enzyme, polymerase enzyme, and next is the ligases. Okay, restriction enzyme, polymerase enzyme we have learned. Next is ligases. Okay, ligases that is functioning as, see from the name itself, it's clear. Ligases, it is ligating or it's joining. So that maintains the integrity. Okay, that maintains the integrity. How it is maintaining that? By joining that breaks in the phosphodiester bond of DNA. We're learning about the structure of DNA. There is phosphodiester bond in DNA. So this helps, this ligases or this ligases, that enzyme helps in the joining of these breaks. Okay, so how these breaks occurs? You know that replication, during replication or recombination. Now we are learning about the recombinant DNA technology. So for recombination of different, there is one host DNA, recombination will happen, right? So for that recombination or for new combinations of DNA fragments, there comes the role. They need to get ligated or they need to join these two fragments of this DNA. So these breaks, there will be some kind of breaks between these two fragments, right? So to join that, this ligases or this ligases enzyme works. It is adding phosphodiester bonds of DNA. That is joining the breaks in the phosphodiester bonds of DNA. So when this breaks comes during replication time and recombination time. Recombination time, now we are learning about the recombination time. So ligases works to join the new fragments of DNA. Next comes vectors. Okay, vectors. Vectors are very important component of recombinant DNA technology or rDNA technology. These vectors, they are the molecules that carries and they can carry the gene of interest. Okay, in the host cell, to the host cell, these vectors, we have told first we will identify the gene of interest, right? After that, we need to transfer that to host cell. So directly we can't transfer these DNA fragment with gene of interest to that host cell. For that, we need the help of some vectors. Vectors are the carriers of this gene of interest, or it is very important in our DNA technology because directly we can't put this DNA fragment into a host cell or host genome. For that, we need the help of vectors. So vectors are the molecular carriers. They are the molecular carriers and these can carry the gene of interest into the host cell. That is the role. The gene of interest is carried to the host cell with the help of vectors. Vectors are actually called as molecular carriers. Okay. And these vectors that can replicate independently without the control of chromosomal DNA. Vectors, to that vector we will attach the gene of interest and will pass them to the host cell. Okay, that's what happening during this recombinant DNA technology. So there the vector is having the capacity for all replication that can replicate independently without the help of the chromosomal DNA, without the control of the chromosomal DNA. So this vector that can independently replicate. Okay. Then now we have learned restriction enzyme, polymerase enzyme, ligase, vectors. So restriction enzyme, first it is making cut, then polymerase enzyme for copy, then ligase enzyme again for joining, recombination, then vectors acting as molecular carriers. With the help of vectors, it is passing to the host organism. Now finally it comes the host organism. So this recombinant DNA, with the help of vectors, whatever we made, that recombinant DNA is introduced into the host organism. There should be a host organism to receive this recombinant DNA, right? There should be a place for this recombinant DNA that should go into a host organism. So this host that is helping in the engineering of this vector. How it is helping? That recombinant DNA, that host organism is helping for this with that desired DNA, for the combination of that genome with the desired DNA. So all these, it is helping, all these processes, all these are taking place with the help of enzyme. Okay, all these enzymes are there. With the help of all these enzymes, these processes are taking. First, restriction enzyme, it is having a specific role. Then polymerase enzyme. Then for combining both ligase enzyme, then it is introduced into a vector. With the help of that vector, it is coming to the host organism. Okay. It will be more clear when we are explaining about the application level. So then it will be clear. With the help of example, we can explain this. Now, let's see restriction enzymes. So in 1963, two enzymes were isolated. Okay, these enzymes, both of these enzymes were able to restrict the growth of bacteriophage. Okay, in 1963, two enzymes were isolated, and both of these enzymes, they were restricting, or they were restricting the growth of bacteriophage. What is bacteriophage? Bacteriophage means that's a type of virus that infects bacteria. Okay, bacteriophage is a type of virus that infects bacteria. So when these two enzymes, from the word itself it's clear, bacteriophage means bacteria eater. So bacteriophage, when it is entering to a host cell, that destroys the host cell. Okay, when this bacteriophage is entering into the host cell, that is destroying that particular host cell. So here, when that bacteriophage is entering to this E. coli, that destroys that E. coli. Okay, but when the enzyme which were isolated were able to, these enzyme, we said about the two enzymes, these enzymes which are isolated, those enzymes were able to restrict the growth of that bacteriophage. Okay, they were able to stop the growth of the bacteriophage. So among these two enzymes, one type, like we discussed about two enzymes, right? Among this, one type added methyl group to DNA. One type of enzyme which is isolated that added methyl group to DNA. The second one, or the other one, that is able to cut DNA. So this enzyme which is cutting, or which was able to cut the DNA, that is called as restriction endonuclease. Okay, that is called as restriction endonuclease. So the enzyme which was able to cut the DNA, that was called as restriction endonuclease. Okay, then HindD2, that is the first restriction endonuclease. Note down which is the first restriction endonuclease, HindD2. You should read it as HindD2. That is the first restriction endonuclease. And later studies prove that this HindD2 that recognizes a specific sequence of 6 base pair. Okay, this HindD2 that can recognize a specific sequence of 6 base pair, and at that particular point, this HindD2 cut double-stranded DNA. Okay, this HindD2, it is able to identify a specific sequence of 6 base pairs. Okay, so that is the identifying base pair of HindD2. For each enzyme, there will be specific sequence. Okay, specific identifying sequence or recognition sequence. So for HindD2, it will be 1, and for another enzyme, it will be another 1. So that recognition sequence for HindD2, that was a 6 base pair sequence. Okay, so at that particular point where that 6 base pair sequence, which is specific for HindD2, there that HindD2 enzyme makes cut in that double-stranded DNA. If there is any other sequence, HindD2 can't identify and it cannot make cut. When the 6 base pair particular recognition sequence, which can recognize HindD2, which can be recognized by HindD2, at that particular point or at that particular site, HindD2 may cut in the double-stranded DNA. Okay, don't get confused. Only at that particular point, HindD2 can recognize on that particular base pair, 6 base pairs. That sequence, that specific sequence only that HindD2 can recognize, and there only it can make cut. Okay, so that 6 base pair specific sequence, that just now I said HindD2 can identify 6 base pair specific a particular sequence. So that particular sequence, that is called as recognition sequence for HindD2. HindD2 can identify that 6 base pairs specific sequence, and there it can make cut. So that 6 base pair specific sequence is called as recognition sequence for HindD2, not for other enzyme, only for HindD2. Okay. Then more than 900 restriction enzymes have been isolated from different strains of bacteria. Okay. All these restriction enzymes, they are getting isolated from different types of bacteria. So around 900 restriction enzymes have been isolated from different types of or different strains of bacteria. Okay, and these restriction enzymes that can recognize different recognition sequence for cutting. Okay, all these 900 enzymes won't be identifying a single base pair or the single base sequence. Okay. That 6 base pair sequence that is specific for HindD2. Like that, for other enzymes, there will be specific sequences. Not this sequence is not for all the enzymes. It is specific recognition sequence for HindD2. Like that, around 900 restriction enzymes have been isolated over 230 strains. Over 230 strains of bacteria. So for each isolated enzyme, it will be having specific recognition sequence. Okay. Then about the naming of these restriction enzymes. How to name this? We said around 900 enzymes are there. So how to name these enzymes? For everything we can't give the same name, right? So how to name? So first letter, the naming of this restriction enzyme, in that the first letter is the genus name. Okay, genus name, and second 2 letters from the species name of that strain from which this enzyme is isolated. Okay, so first name is from the genus name, first letter is the genus name, and second 2 letters that are from the species. Okay, first from the genus and second 2 letters are from the species of that particular strain from which it is isolated. Okay, for example here, Echo R1. So in that name, how it came, this name? That name came from Escherichia coli. The first letter, first is R. The letter, that name came from Escherichia coli RY30. Okay. From this strain, this eco R1 isolated. The eco R1, this enzyme got isolated from Escherichia coli RY13 from this strain. Okay. So R letter from the strain name. Okay. The eco, from here you can see R letter from the strain name, and the Roman letter, Roman number, this letter R, this is from the strain, and the Roman letter 1, R1 is, that one is from, that is the order in which that enzyme is isolated from that strain. From this strain, they might have isolated many enzymes. So that is the order in which that enzyme is isolated. That is the first isolated enzyme means they'll name it as eco R1. If the second isolated one means they will give the eco R2, Roman letter 2. That is second isolated enzyme, like that. Okay. So first letter eco R1, that comes from Escherichia coli RY13. Note down this. These are all important question for NEET exam. Eco R1 comes from Escherichia coli RY13. Okay. In that eco R1, the letter R is from the strain RY. So that R is from strain. And the Roman number R1. After R1, Roman letter 1 is there. That Roman number. What that indicates? That indicates the order in which the enzymes were isolated from that strain of bacteria. So from this strain, that is the Roman number, that shows the order in which that enzyme is isolated. So that's how the naming is done. How the naming of that enzyme is done? First letter of the name comes from the genus. First letter from the genus name. Here, while writing the scientific name, first you will write the genus name. Secondly, the species name. Right? So the genus name, first letter comes from the genus, and the second two letters comes from the species. So here you can see first letter, which is the genus name Escherichia coli. In this Escherichia, that is the genus name, and coli is the species name. So first letter is from the genus E, from Escherichia. From that we took the first letter E. Then coli, that is the species name. First is the genus, then the species. So in that species, the next two letters, second two letters comes from the species. So CO, coli, from coli we took CO. Eco, then R. Letter R is derived from the strain name. Here strain name RY13. That is the strain name RY13. So from that strain we took the letter R. Then Roman number 1 indicates the order in which that enzyme is isolated. Okay. So that's how we are naming the enzyme. Next, restriction enzymes. Now we learned about the naming of restriction enzyme. Now let's learn some more points about restriction enzyme. This restriction enzyme that belongs to nucleases. Okay. Which group it belongs to? Nucleases. Restriction enzymes belongs to nucleases, and that is a large class of enzyme. Restriction enzyme belongs to the large class of enzyme called nucleases. Okay. So nucleases, that is a very large class of enzyme, and restriction enzyme belongs to that nucleases class. Okay. And this restriction enzyme, they are of two types: restriction exonuclease and restriction endonuclease. Okay. Restriction exonucleases and restriction endonucleases. Okay. Restriction enzymes, they are belonging to the large class which is called as nucleases, and it is of two types. Restriction enzymes, they are of two types, restriction exonucleases and restriction endonucleases. This exonucleases that remove nucleotide, exonucleases, what it will do? That removes nucleotide from the end of the DNA. From the end of that DNA, that removes the nucleotide. Okay, which one? Exonucleases. Exonucleases remove the nucleotide from the end of the DNA. At the same time, what endonucleases will do? Endonucleases cuts DNA at specific positions that will identify that recognition sequence, and it will cut the DNA at specific positions. So there are two types of restriction enzymes, restriction exonucleases and restriction endonucleases. Restriction exonucleases remove nucleotides, that remove nucleotides from the end of the DNA. At the end of the DNA, nucleotides are present, means this exonucleases will remove that nucleotides. But what endonucleases will do? That will identify specific positions within that DNA, and there it makes cuts in the double-stranded DNA. Okay. So this restriction endonuclease that inspects or that checks the length of that DNA sequence. That's how it functions. Okay. So how it is functioning? That restriction endonuclease that is checking the length of the DNA sequence. That checks the length of the DNA sequence, and that's how it works. Okay. Then after checking the length of the DNA sequence, what that will do, this restriction endonuclease, it will find out the specific recognition sequence. We said about if HIN D3 or HIN D2, that is the restriction enzyme which we are using for our work, then it will identify that six base pair specific sequence, which is the identifying or recognizing sequence or recognition site that will get identified by that HIN D2 restriction enzyme. But here we are generally speaking about the restriction enzymes. Each restriction endonuclease first that will identify or first that will check the length of the DNA sequence. So once it is finding the specific recognition sequence, that will bind to that DNA, and then it will start the cutting. Okay, then it will cut the double-stranded DNA, that both the strands, the two strands of the double-stranded DNA, it will cut. Okay, when it makes cut or where it is making that cut? At specific points, only at specific points it is making that cut. Okay, when it is making that specific point, at that point, when it is identifying that recognition site, specific identification sites will be there. So that recognition site, there only it will make cut for both the strands in a double-stranded DNA. So in specific points in the sugar phosphate backbone, there it makes cut. You know the structure of DNA, right? So in the sugar phosphate backbone, it makes cuts. Okay. Then each restriction endonuclease that recognizes a specific palindromic nucleotide sequence in the DNA. A specific nucleotide sequence that will be in palindromic form. Okay. So here you can see a restriction enzyme, how it is working. Here, the restriction enzyme, it is cutting. It is making cut in both the strands. and both the sides it is making ends that I'll explain. Then it is combining both strands. Okay, this restriction enzyme EcoR1 here. We're showing the enzyme EcoR1, the restriction enzyme EcoR1 that is cutting both the strands, and here the recombination is taking place. Okay, I'll explain that in detail. So restriction enzyme, it is belonging to larger class of enzyme that is called as nuclease, and there are two types, exonuclease and endonuclease. This exonuclease removes nucleotide from the end of the DNA, but endonuclease, what it is doing, it is making cuts at specific positions in that DNA. That specific positions are called as recognition sites. So each restriction endonuclease, first that will find out the length of that DNA sequence, and in that whole DNA where it is finding specific recognition sequence, there it will bind, and then it is making cuts in the double-stranded DNA. Okay. In the sugar phosphate backbone, it is making cuts. Okay. Then each restriction endonuclease that recognizes a specific palindromic nucleotide sequence in the DNA. We said about the recognition site, that is where it is identifying a specific palindromic nucleotide sequence in the DNA. Okay. Here, just now we said about palindromes. What are palindromes? So what is palindromic sequence? That means the group of letters. You can see a group of letters that can letters can form the same word. So if you are reading from both directions, from left to right or right to left, in both directions it will give the same word. Okay, in both directions we are reading also, it will give the same word. Here we have given an example, Malayalam. The word Malayalam, you can read, see, M-A-L-A-Y-A-L-A-M. Right? From left to right we are reading, means these are the letters, M-A-L-A-Y-A-L-A-M. This is how we can read from left to right. See, if you are reading in opposite direction, from right to left, M-A-L-A-Y-A-L-A-M. It is giving the same letters, and here from right to left also you can read the same word Malayalam. Right? It is giving the same letters in both directions. So you can, that forms the same word when we are reading in both directions. Okay. Such group of letters which are giving the same word when we are reading in both directions, when it is read from back forward or backward direction, when it is read in forward direction or backward direction, that group of letters forms the same word. Such group of letters are called as palindromes or palindromic sequence. Palindromes. Here, palindromic sequence means in a DNA, the base pair, here base pairs are present in the DNA, right? Like A, T, G, C, such kind of base pairs are present. So when that base pair, that group of base pairs, when we are reading that on the strands, in both directions you can read and you can get the same sequence. So in the case of sequence, you can see if you are reading here, this is the sequence G-A-A-T-T-C. This is the sequence when you are reading from 5' to 3' direction, right? And here in 3' to 5' direction, if you are reading also C-T-T-A-A-G. Okay, so if you are reading in the two strands, one is 5' to 3' direction and another one 3' to 5' direction. Okay. So if the complementary strands, when the complementary strands comes also, it will be like this only. Okay, that is called as palindromic sequence in DNA. So it is giving when we are reading from forward, when we are reading in forward direction or backward direction, that group will give the same word. That is called as palindromes. Okay, so in the case of sequence, you can see reading from 5' to 3' direction or 3' to 5' direction, it gives the same. Okay, that is called as palindromic sequence. Got it? Do you have any confusion here? GAATTC. Okay, when we are reading GAATTC, from opposite direction you are reading also. Okay, so that comes in the double-stranded DNA. It will become more clear when we are explaining. So later it is coming all these things. So that time if we are explaining, it will be clear properly. So next, what we are going to learn is how this restriction enzyme cuts. Restriction enzyme is used for cutting, right? So how it is cutting? Let's see how this restriction enzyme is helping in cutting. Okay. So this restriction enzyme that is cutting the double-stranded DNA. Here you can see the double-stranded DNA. So restriction enzyme that is making cutting in the double-stranded DNA. Okay. So where it is making that cutting? It is making cutting little away from that palindrome site. Palindrome sequence. That is the recognition sequence of that particular restriction enzyme. So not exactly at that point it is making cutting. It is making cutting a little away from that palindromic sequence, which is the recognition sequence. Okay, but on both strands, between the same two bases, this enzyme makes cuts. Okay, on both strands, on double-stranded DNA, on both strands, between the same two bases, like palindromic sequences that will identify. So on both strands, it will make cut between the same two bases. There won't be any changes. On the same two bases, it will make cut. This enzyme will make cut on the same two bases. Okay. So that makes single-stranded portion at the end of that. See here you can see. In the figure you can see at the same side, the enzyme cuts both DNA strands. Here this is the vector DNA, this is the foreign DNA. Here, this is the double-stranded form, right? Here at the end, we have given the sequence of that double strand. Here also they have given the sequence. Okay, this is the palindromic sequence. You can check this palindromic sequence on both strands. So there the restriction enzyme is identifying, or that is the recognition site of that restriction enzyme, right? Echo R1, that is the recognition site of these enzymes. So there it identifies, and there it makes little away from that. It is making cut. See here, it is making cut between G and A. And in this, in the foreign DNA strand also, it is making cut between G and A. Here also, in the opposite strand also, here, it is making cut in the same way. Got it? So what is happening after that? You can see palindromic sequence at both sides, and on this echo R1, that is the restriction enzyme that works on that acts and that makes cut at the DNA strands. Right? Here it is making cut at the DNA strands. So after cutting, you can see the overhanging stretches. Here you can see the overhanging stretches of DNA strands. Okay, the overhanging stretches of DNA strands after cutting you can see. And in the first picture, down you can see. In the second one, top you can see. So this is called as, these ends are called as sticky ends. Okay, after cutting that overhanging structures, you can see overhanging structures of DNA, that is called as sticky ends. Why it is called as sticky ends? Because these ends that forms the hydrogen bonds. Here you can see the base pairs, right? Here you can see the base pairs, and from here it is not there. It is single strand. Up to here it is double strand, and this part is cut now. So you can see here base pairs, and there is no complementary strand. In this stage also, in this side also you can see there is no complementary strand here. So why it is called a sticky end? That makes that end or these end forms hydrogen bond with the complementary cut part. That means when both these sticky ends of DNA fragments are joining, then that forms the hydrogen bonds. Here you can see base pairs. There is no complementary strand here on the opposite side, and here also there is no other strand. So when these, see, this is the DNA fragments which we need to join. This is the vector DNA. With the help of this vector DNA, we are joining the foreign DNA. This DNA is having the desired gene. That foreign DNA we need to join along with the vector DNA. Then only we can pass this whole DNA into a host organism for the growth, right? So when we are combining or recombining this DNA of that vector DNA and the foreign DNA, we are joining this. So while joining these sticky ends, there it forms the hydrogen bonds between the base pairs. Okay, so DNA fragments join at sticky ends when we are making recombinant DNA. Okay, so here you can see it is joined. So this is the recombinant DNA. There is a new combination. This is the recombinant DNA. Okay, there is a vector DNA and there is a desired gene or a foreign DNA which is having the desired DNA. Both are combined. This is the recombinant DNA. Got it? So the fragments join at sticky ends. So here you can see it can't join simply, right? There also the role of another enzyme comes. Here comes the role of ligase enzyme. The ligase enzyme, it is for joining purpose, or the role of that ligase enzyme we have already told, joining. That is the role of ligase enzyme. That's why it is called as molecular glue. Ligase enzyme is called as molecular glue because it is helping for joining the DNA. So that stickiness of these ends that facilitates the action of DNA ligase enzyme. DNA ligase enzyme which helps in the joining of DNA. DNA ligase, that enzyme acts here and that helps in the joining of that DNA, and which is helping in the action of ligase enzyme, these sticky ends that provides the situation for acting this ligase enzyme. Due to that sticky ends, this ligase enzyme can work there and that can join and that can form a recombinant DNA or a new DNA. Okay. Here, in genetic engineering, this restriction enzyme, that means restriction endonuclease, that is used for making recombinant DNA molecule. Okay, restriction endonuclease. We said it's mainly used for cutting DNA, double-stranded DNA. So it's mainly used in genetic engineering. The main role of restriction endonuclease is for making recombinant DNA molecule. There comes the role of this restriction endonuclease. That means the combination of two different DNA molecules which are from two different sources. Here you can see there is a foreign DNA and a vector DNA. From both of these, that restriction enzyme that will cut the foreign DNA and that vector DNA at specific point, right? Here, restriction enzyme is making small pieces of that foreign DNA which is having the desired gene. At the same time, the vector DNA, it is making some cuts, right? Then comes ligase. Ligase that joins the a foreign DNA. Here the foreign DNA and the vector DNA both are getting joined with the help of ligase enzyme, or that is called as ligase, which is the molecular glue. Ligase enzyme is otherwise called as molecular glue. Okay. Then this is the recombinant DNA. After joining, these both will join and it will form a complete structure. So that is the recombinant DNA molecule. Okay, with the help of that vector, here the desired DNA also desired gene, that DNA which is having the desired gene, that will get joined with this vector, and then it is transferred into a host organism. Okay. Here inside the host organism it will get transferred. Okay. So here when it is using for the, when we are using that same restriction enzyme, for what purpose? The combination of two different DNA molecules which are from different sources or different genomes. To combine that we are using, in genetic engineering, for that purpose we are using the restriction endonuclease. Here when we are using the same restriction enzyme for cutting both the foreign DNA and the vector DNA at specific point, after recognizing that palindromic sequence and that resulting the DNA fragments that will be having same kind of sticky ends. See the foreign DNA and the vector DNA, both, in both we are using the restriction endonuclease, the same restriction endonuclease to cut, right? So that makes similar sticky ends, same kinds of sticky ends, and that helps in the joining. Here you can see in the previous figure you can make out both echo R1, that is the enzyme, restriction enzyme which we have used. So that is identifying that palindromic sequences, and there it makes cuts, right? In the foreign DNA and in the vector DNA, in both. Here this is the vector DNA, there also it makes cut, and in the sticky ends which is formed in the foreign DNA, there also that only made the same echo R1 that only made the cut, right? The same restriction enzyme. So that makes similar sticky ends, and that helps in the joining. These both similar sticky ends that can be joined, end-to-end joining with the help of ligase enzyme. Got it? So that's how recombinant DNAs are forming. This is the process or procedure of or stepwise process of recombinant DNA molecule formation. Okay, that's how recombinant DNA molecules are forming. Got it? Is it clear? Do you have any doubt? Okay, so these are the main techniques which we are using in the formation of rDNA molecule. So later we can learn about the separation and isolation of this DNA fragments and all that we'll learn in the next class. So go through all these portions. If you have any doubt, you can ask me in the next class. Thank you.